As you know, each species is characterized by a particular number of chromosomes. This chromosome set or genome in higher plants or animals is usually represented once in their haploid cells, for example, gametes, spores, etc., and twice in their diploid somatic cells. This module will deal with the genetic effects of numerical chromosome abnormalities. Of the two classes of chromosome abnormalities, change in chromosome number is more frequently observed among the live born children. A numerical change is also cytologically the easiest to observe. Basically, there are two types of numerical changes in the cell chromosome number. These are euploidy, in which there is an addition or deletion of entire set or sets of chromosomes, and aneuploidy, which involves the addition or deletion of individual chromosomes within a set. Both euploidy and aneuploidy thus alter gene doses in the affected individuals. In this module, we will examine the numerical chromosomal aberrations in details. There are basically two types. One is euploidy. The term euploidy refers to the condition when a complete set or sets of chromosomes are present in a eukaryotic organism or in its gametes. When one complete set of chromosomes is present, the condition is called monoploidy, and if two complete sets are present, it is termed diploidy 2n, and when more than two sets of chromosomes are present, it is called polyploidy. Let us discuss these one by one in the following subsections. First is monoploidy, is also referred as haploidy, and you already know that the n or the one set chromosome number is usual for gametes of diploid animals, but is unusual for their somatic cells. Monoploidy is seldom seen in animals, but is found in male honeybees, ants and other insects, in which male haploid develops from unfertilized eggs, a phenomenon known as parthenogenesis. In contrast, plants have a gametophyte stage in their life cycle that is characterized by reduced, that is n chromosome number. In higher plants, this state is however brief and inconspicuous. In some lower plants, for example, algae, bryophytes, the haploid or monoploid phase constitutes the dominant part of the life cycle. The monoploid plants can be recognized in natural population by observation and verification through cytological preparations. These plants are usually frail in appearance and have small leaves, low viability and high degree of sterility. Sterility is largely because of irregularities at meiosis. Since only one set of chromosome is present, therefore, no pairing is possible, and if the meiotic process succeeds at all, the dispersal of chromosomes to the poles is irregular and the resulting gametes are highly unviable. The monoploids are valuable experimental tools because they undergo no segregation and carry a single set of genes. Polyploidy, the euploids with three or more complete sets of chromosomes or genomes are called polyploids. More especially, the polyploid organisms can be classified as triploids 3x with 3 sets of chromosomes, tetraploids 4x with 4 sets of chromosomes, pentaploids 5x with 5 sets of chromosomes, and so on. Polyploidy results due to errors during meiosis or fertilization. There are 4 ways of origin of polyploid condition. First, double fertilization of an egg by 2 sperms, that is dispermy, or fusion of an egg, a polar body and a sperm cells results in triploidy. Second, an unreduced diploid or 2n gamete uniting with a normal haploid or n gamete also produces triploidy. Third, many a time tetraploidy occurs after fertilization due to a mitotic error. 2 cycles of chromosomal replication, that is endo reduplication without cell division, result in a tetraploid cell. Fourth, fusion of 2 diploid cells is another process whereby tetraploidy may result. In human beings, nearly all triploid and tetraploid fetuses are spontaneously aborted or stillborn. Polyploidy is rather common in plant kingdom, but is rare in bisexual animals. Most animals have well defined separate sexes characterized by the presence of specific sex chromosomes. Multiple chromosome sets lead to variation in the number of the sex chromosomes, for example, XXXY or XYYY, a situation incompatible with the normal sex determining mechanism. As a result, the animal polyploids are restricted to group that reproduce asexually or are hermaphroditic or parthenogenetic. Examples are earthworms, some types of shrimp, and the parthenogenetic species among the insects and lizards. Polyploidy has played a significant role in the evolution of plants. Polyploidy is possible in plants because of the general absence of sex chromosome and a large number of plant species being monoecious. Plants have an additional feature, that is reproduction by self fertilization or by asexual means, which enables the partially sterile polyploid species to propagate. The examples include many mosses and ferns and a few gymnosperms and almost half the angiosperms. New polyploid varieties of cultivated and ornamental plants are constantly developed and introduced by the plant breeders. Some examples of important polyploid crop plants are apple, banana, barley, coffee, cotton, peanut, potato, strawberry, sugarcane, watermelon, tobacco, wheat, and many ornamentals. The phenotypic effects of polyploidy are generally those of increased vigor, growth and size, hardiness, ability to occupy less favorable habitats, and overall superiority as compared to their normal counterparts. One reason for such superiority is the fact that the expression of any deleterious or lethal allele can be effectively masked by the activity of the two or more normal alleles that are present in the genotype. However, because of meiotic irregularities, polyploidy is often accompanied by partial or complete sterility caused by non-functional gametes, particularly the male gametes. Autopolyploidy and allopolyploidy. We have classified the polyploids on the basis of the number of sets of chromosomes present. We can also divide them into two general groups according to the source of their chromosome sets. Polyploidy derived from multiplication of a single ancestral set of chromosomes is known as autopolyploidy. Usually, it results from the fusion of unreduced gametes. However, in many cases, the polyploid species have complete sets of chromosomes from two or more different ancestral species. Such polyploids are known as allopolyploids, and they originate from occasional hybrids that occur between distinct diploid species. Let us examine these two states in detail. The presence of varying numbers of quadrivalents, that is 4 homologous chromosomes instead of the usual 2 pairing with one another during meiosis, suggests autopolyploidy. The unequal segregation of chromosomes in the quadrivalents is the main reason why the autopolyploids are sterile of varying degrees. Chrysanthemum is well known example of autopolyploidy. In this genus, the basic number is 9. Its polyploid species are known to have 18, 36, 54, 72 and 90 chromosomes, all multiples of 9. Potato or Solanum tuberosum is another example. Its basic chromosome number is 12. Its autopolyploid forms with chromosome numbers 24, 36, 48, 60, 72, 96, 108, 120 and even 144 are available. Allopolyploidy is the most common natural form of polyploidy that has occurred in various plant groups. Many of the present day plants have resulted from this kind of hybridization. The polyploid species that are established in nature have genomes that correspond more or less completely to the combined chromosome complements to two different but related diploid plants. In the allopolyploids, since two homologs of each specific chromosome are present, meiosis may occur normally and fertilization may successfully propagate the tetraploid sexually. These allopolyploids, also known as allotetraploids or amphiploids, have undergone hybridization somewhere in their ancestral history. Allopolyploidy thus represent a method by which new species may be formed almost immediately, whereas the autopolyploids result in meiotic anomalies and dead ends with reference to evolution. Cultivated wheat is an excellent example of allopolyploidy. Cultivated bred wheat is a hexaploid with 42 chromosomes constituting complete diploid genome of 14 chromosomes from each of the three ancestral species. The 42 chromosome allopolyploid is thought to have originated through hybridization. Another allopolyploid, Raphanobrassica, was experimentally synthesized by G. D. Karpyshenko by crossing two common vegetables that belong to different genera. These are radish, Raphanus sativus, and cabbage, Brassica oleracea. Both the species have 9 pairs of chromosomes. The diploid hybrid has 18 chromosomes, 9 from each parent, but is sterile and cannot perpetuate itself, largely because of failure of pairing between the unlike chromosomes in meiosis. When the chromosomes of the F1 hybrid were doubled, a fertile polyploid named Raphanobrassica was produced with 18 radish and 18 cabbage chromosomes. Because two sets of chromosomes were now present from each parent, pairing was quite regular and hence normal gametes were produced. This experiment suggested the possibility of incorporating desirable genotypes from two different species into a new polyploid species. Induced polyploidy. Polyploids may arise naturally or may be artificially induced. Application of the alkaloid colchicine derived from the autumn crocus Colchicum autumnale disturbs spindle formation. The cell division proceeds as usual, but since the spindle formation is inhibited, the cytoplasmic phase of cell division does not occur. Instead, restitution nuclei with the double number of chromosomes are produced in the treated tissue. Subsequent nuclear divisions are normal, so that the polyploid cells line once initiated is maintained. By crossing an autotetraploid with a diploid one, triploid can be produced as in watermelon that are partially seedless. Applications of polyploidy. For example, bread wheat. The genus Triticum consists of polyploid series of wheat. The diploid species T. monococcum or encorn wheat 2n is equal to 14 were the first to be used for food about 10,000 to 12,000 years ago, about the time civilization began. At that time, seeds of wild plants were collected for food and subsequently brought in cultivation. The encorn wheat is hulled and is small as compared to its polyploid relatives. The emmer and durum wheat belong to the tetraploid species T. turgidum 2n is equal to 18. These wheat originated from a naturally occurring wild tetraploid variety whose grains are larger than the encorn wheat. The emmer wheat is hulled, but the durum wheat is particularly desirable because of its free threshing quality, that is, the hull is easily removed by the threshing, and unlike hulled wheat, it does not need to be ground for human consumption. Varieties of bread wheat Triticum aestivum are agriculturally the most important. Bread wheat is hexaploid 2n is equal to 42 derived from hybridization of tetraploid emmer wheat with a diploid relative, the wild grass Agelops squarosa 2n is equal to 14. Bread wheat is especially desirable because of its free threshing and has large grains, and also has a high content of proteins called the gluten's. Polyploid fruits and flowers. Many varieties of plants that fulfill human needs have been found to be polyploids. The polyploids were selected for cultivation because of their large size, vigor and ornamental value. In Macintosh apple trees, some twigs produce extra large fruits. These twigs were known as giant spots and were found to be tetraploid 4n. These giant spots were propagated into whole trees which produced extra large fruits. Similar spots with giant fruits are also seen in barlett pears, several varieties of grapes and cranberries. These giant spots develop naturally, but with colchicine treatment a number of polyploids have been developed artificially. This technique has provided a way to explore the mechanism involved in the polyploid formation. The triploid watermelons with 33 chromosomes have great market potential because they are sterile and lack seeds. Several tetraploid flower varieties such as marigolds and snapdragons are widely cultivated. Aneuploidy. The aneuploids are individuals with unbalanced chromosome sets, that is with extra or missing chromosomes. Their chromosome complement contains at least one more or one less chromosome than the normal diploid number. If one homologue of the normally diploid complement is present 3 times, the condition is called trisomy, and an individual of this type is called trisomic, and the condition can be designated by the symbol 2n plus 1. Tetrasomy 2n plus 2 indicates that one chromosome of the complement is present 4 times. A 2n plus 1 individual would be doubly trisomic for 2 different homologues, and the 2n plus 2 individuals would be doubly tetrasomic and so forth. The reciprocal situation involving loss of a chromosome from a pair is called monosomy 2n minus 1, and loss of a whole pair of chromosome is known as nullisomy 2n minus 2. Non-disjunction of homologues during meiosis. Non-disjunction is the failure of chromosomes to separate during cell division. If one pair of homologues fail to disjoin, then n plus 1 and n minus 1 gametes will result, and if these gametes are fertilized with the normal gamete, then 2n plus 1 and 2n minus 1 individuals would be formed instead of normal 2n. The chromosome constitution of the zygote is critical to normal development, because any chromosome abnormality present in the zygote will be passed on through each succeeding generation of cells. The single cell line produced by meiotic non-disjunction. All forms of aneuploidy have serious consequences in meiosis. A trisomy in Jimson weed. The Jimson weed, the Datura stramonium, shows a considerable amount of morphological variation in many traits, particularly the fruits. The normal chromosome number of this plant is 2n is equal to 24. Blacksley and Belling in 1924, who studied this system extensively, showed that each of the particular morphological variants had 25 chromosomes. In each variant, one of the 12 kinds of chromosomes was found to be in triplicate, that is, the somatic cells were 2n plus 1, and it is a case of trisomy. These people thought, since the Jimson weed has 12 pairs of chromosomes, 12 recognizable trisomic should be possible, and as expected, they could collect all variants for the 12 chromosomes. Autosomal aneuploidies in man. After studying what is aneuploidy, its origin and classic example of Jimson weed, let us now take some commonly found syndromes due to autosomal aneuploidy in man. The most common aneuploid condition seen with appreciable frequency in live born children are trisomics 13, 18 and 21. In order of increasing frequencies, these trisomics are Patau, Edward and Down syndromes respectively. Trisomy 21 Down syndrome. It is one of the well known and best studied examples of chromosome caused abnormalities in human beings. This condition, which is an autosomal trisomy, was first described by Langdon Down in 1866 and was originally called Mongoloid idiocity or Mongolism. This syndrome is the result of an additional chromosome 21, one of the G group, and is designated as 47/21 plus. This trisomy is found in about one infant in every 600 to 700 live births. The Down syndrome DS phenotype. Many phenotypic characteristics have been associated with Down syndrome. A few dozen of these traits are present in most children suffering from this disorder, and many of them appear as siblings at a first glance. A child with DS ages rapidly, has an IQ range of 25 to 70 and has a flat facial profile and a broad saddle nose. The ears are small and round, the tongue is large and flat, the child has protruding lower lip, a broad short skull, thick short hand with stubby fingers and thick short feet and body. Origin of Down syndrome. One of the main ways in which this trisomy condition originates is through non-disjunction of chromosome 21 during meiosis. Failure of paired homologs to disjoin during anaphase first and second can result in the male or female gametes with the n+1 chromosome composition. Following fertilization with the normal gamete, the trisomic condition results. Trisomic 18 Edward syndrome. In 1960, John Edward and his colleagues reported an infant trisomic for a chromosome in the E group, now known to be chromosome 18. It is designated as 47, 18 plus. Incidence is about 1 in 4,000 live births. 80% of trisomy 18 cases are due to meiotic non-disjunction. 10% are due to translocations and 10% are the result of trisomy mosaics. Characteristics of trisomy 18 children are highly variable and pronounced. They demonstrate severe mental retardation, odd shaped head, webbed neck, short thumb, low set ears, small mouth, receding chin, the index finger overlapping the middle and ring fingers, rocker bottom feet and clefts in the lip and palate. Heart defects are found in nearly all trisomy 18 births as well. Survival time is less than 4 months on the average. Death is usually caused by pneumonia or heart failure. Trisomy 13 Patau syndrome. In 1960s, Klaus Patau and his associates observed an infant with gross developmental malformation with a karyotype of 47 chromosome. The additional chromosome was medium sized, one of the acrocentric D group. The G and Q banding techniques have identified the extra D chromosome as number 13. The trisomy 13 condition has since been described in many newborns and is called Patau syndrome, being designated as 47/13 plus. 70% of the trisomy 13 cases are caused by meiotic non-disjunction, 10% of the cases are trisomy mosaics and 20% are partially trisomic due to translocation. As the chromosome 13 contains considerable active genetic material, most chromosome 13 trisomics abort. Of those that are live born, 90% die during their first year of life. Those few that survive beyond the age of 2 years suffer from severe mental retardation. The characteristic of trisomy 13 include seizures, moderate microcephaly, small head, wide spaced eyes, beak nose, clefts on the lip and palate, low set ears, scalp defects and characteristic trigger thumb, that is the thumb and index fingers overlap the third finger. Trisomy 13 babies display a simian crease, one continuous crease across the palm of the hand as in Down syndrome, as well as polydactyly that is extra fingers and toes, congenital heart disease, kidney disorders and malformed genitalia. Sex chromosome aneuploidies. Aberrations in the number of X and Y chromosomes are more readily found among humans than are the aberrations in the number of autosomes. In this section, you would study two kinds of aneuploidy in 6 chromosomes and their effects. XXY-Klinefelter Syndrome. An extra X chromosome in addition to the usual male XY chromosome complement 47 XXY has been associated with a male phenotype, the Klinefelter syndrome. It was described by H. F. Klinefelter and his colleagues in 1942. It is estimated to occur in 1 per 500 live male births. The risk of a Klinefelter birth is associated with increasing maternal age. It is believed that the XXY constitution originates either by fertilization of an exception XX egg by a Y sperm or of an X egg by an exceptional XY sperm. The Klinefelter syndrome patients are always male by virtue of having Y chromosome, but they have variable male genital development. The characteristics of Klinefelter syndrome male are underdeveloped testes, hypogonadism causing sterility, have small prostate glands and a tendency towards femaleness, particularly the secondary sex characteristic. They show some female features such as breast development after puberty, gynecomastia, pubic hair and distribution of fat. Unlike adult males, they lack facial hair. Most of them demonstrate mild to severe retardation. Many adjust poorly to breast development and exhibit emotional problems. The Klinefelter syndrome patient is one of the most amenable to treatment using hormones and surgery. XO Turner syndrome, a condition XO exemplify monosomy of the X chromosome in humans. This condition is termed as Turner syndrome and was first described by Turner in 1938. These people are female because of the absence of Y chromosome. They have a genomic complement of 45, X indicating that they have a total of 45 chromosomes, in contrast to the normal 46. The sex chromosome complement consists of 1 X chromosome. Turner syndrome is the least frequent of the live born sex chromosome aneuploidies, as 98% of the XO conceptions are spontaneously aborted, yet it has a frequency of about 1 in 5,000 live born infants. About 20% of all spontaneous abortions are 45 XO, making this one of the most common chromosome aneuploidies known. The characteristic feature of an adult with Turner syndrome are relatively short stature due to failure of the ovaries to develop, webbed neck, low set ears, wide spaced and underdeveloped breasts, sparse pubic hair, abnormal hormone levels, narrowing of the aorta and failure to menstruate. Amenorrhea. In conclusion, in this module you have learned that abnormalities in chromosome number can either involve entire set of chromosomes or individual chromosomes. When complete sets of chromosomes are added to the diploid genome, a polyploid is created. The sets may have identical or diverse genetic origin, creating either autopolyploidy or allopolyploidy respectively. Aneuploidy is the gain or loss of one or more chromosomes from the diploid content. The major mechanism of development of aneuploidy is non-disjunction. Many kinds of aneuploidy leading to abnormal conditions are known to occur in man.